Hello everyone, welcome back to another class on regeneration biology. So in today's class, we will learn more in detail about the Wnt signaling. Different extracellular or different cellular signaling events need to be studied and addressed, which we will do in the subsequent classes. And in today's class, we will try to understand how Wnt signaling influence different tissue types, how and why a Wnt signaling becomes pivotal in a regeneration event. And we also see some examples. Wnt signaling is not that welcome for a regeneration of some tissues. And here, this is not exclusively meant for zebrafish, because zebrafish we have seen Wnt signaling is an important signaling event for the retina regeneration. Like in the previous classes, also we have studied that Wnt signaling, if you stabilize the beta catenin, it can allow a regenerative response even without any injury at all. So we will study how other organisms respond to Wnt signaling. So importance of Wnt signaling during tissue regeneration has been well characterized. And regenerative medicine is a translational field of that combines different aspects of cell biology and molecular biology and tissue engineering, and it constructs spare organs that help to injured or a defective tissues, or to generate or restore a damaged portion and restore the normal function. Say for example, a person who got a myocardial infarction and the blood supply to his heart muscle is stopped, and this often leads to some scar formation after healing. And this scar is not welcome because it will not contract. For heart to function, the wall must contract. So this scar will be a hindrance in the contract. It is just like, you are chewing something, some nice food, and I put a stone in between. So you cannot break, you cannot chew the stone, and it will stop the smooth grinding of your existing food. So contraction has to happen perfectly for the heart muscle to function. So if you can get rid of this scar by some means, that itself is a welcome sign. You need not make a heart muscle restored back, but at least you get rid of the scar. That is one of the right approach to perform or to restore a normal heart function, so that the heart can perform in a nice way. So this is particularly important with the specific organs such as heart, central nervous system, retina, or limbs, which possess very limited regenerative capacity, especially in mammals, what we are talking about. Wnt is normally it's a wingless type MMTV integration site family. So it is a larger protein name. Wnt signaling is an evolutionarily conserved system, highly active during embryogenesis. Some researchers point out that there is Wnt-like molecules present even in plants. Animals it is there, but plants also have molecules similar to that of Wnt signaling, and some researchers say that they can cross react also. So I will not dwell in detail, just to tell you that it is an evolutionarily conserved pathway. As such, the regenerative medicine has received peculiar attention in the last few years, and in this regard, the Wnt beta-catenin signaling pathway has been the subject to intensive research. It plays many essential roles regulating progenitor cell fate, developmental decision, proliferation during embryonic development, and adult tissue homeostasis. So the roles of Wnt signaling in various aspects is been well characterized and well studied. So regeneration is nothing but a extension or a recapitulation of regeneration biology. So the Wnt beta-catenin signaling pathway contributes to both stem and cancer stem cell maintenance. So this also another factor which you should keep in mind. During regeneration, a lot of progenitors are induced, but you don't want any cancerous tissue to be formed because of an injury response. Wnt dysregulation and excessive activation during pathological conditions led to morphological and functional changes in the heart, resulting in impaired myocardial regeneration under pathological conditions such as myocardial infarction, heart failure, and coronary artery disease. So Wnt signaling If it is overactive also problem. If it is not acting also is a problem when it comes to regeneration is concerned. So the adequate Wnt signaling response is expected for a perfect regeneration. Several groups of Wnt inhibitors have demonstrated the ability to modulate Wnt pathway, thereby significantly reducing fibrosis and improving cardiac function after myocardial ischemia. Like I told you, if you can get rid of the fibrosis itself is a right approach. Their inhibitory effect can be realized at multiple levels, which include inhibition of Wnt ligands and the inhibition of frizzled receptors and stabilization of the beta-catenin destruction complex and the disruption of the nuclear beta-catenin interaction at different levels. Say for example, I gave you a plate of food and you have to eat, and I can stop you in multiple ways. Either I can put a tape on your face, tie both your hands, or cover the plate, or remove the food from your plate. There are multiple ways in which I can prevent you from eating food. So same way, signaling pathway also you can influence at different phases. The roles of beta-catenin pathway in tissue development and regenerative medicine. So this is a cartoon which explains how Wnt signaling is important during development and also during a tissue healing. So blastocyst, this is the inner cell mass from which this is a developmental stage where the embryonic stem cells can be collected, and the maintenance of pluripotency is necessary for if you want to passage onto the cell. And you can see frizzled receptors are expressed in these cells and they have to be constantly active if they have to be remaining in a stem cell state. So for which people supplement the Wnt3a, that is Wnt3 ligand externally into these cells. And establishment of stem cells, their nature, if it has to be maintained, the GSK3 beta that usually inhibits, because GSK3 beta causes the degradation of the beta-catenin and the Wnt signaling will be turned off. So Wnt has to be expressed by these stem cell if you want to properly maintain. It includes different Wnt's are there, Wnt3, Wnt5, Wnt5b, Wnt7. There are different Wnt ligands are expressed. Some tissue have got more of a given ligand than the other. Since these cells being embryonic stem cells, they have a variety of Wnt ligand that being produced. And when it comes to bone marrow or umbilical cord, both have got some kind of stem cells, bone marrow stem cells or umbilical cord stem cell, and they are often referred to as mesenchymal stem cell. And cell proliferation rate happens at a rapid rate in these cells, and they also have frizzled receptors that is meant for activating the Wnt signaling, as you found with the embryonic stem cell. And their frizzled receptors are expressed in these cells, and Wnt5a, instead of GSK3 beta, Wnt5a is acting as a negative regulator, so they plays an inhibitory role, but just Wnt5a. And the maintenance of pluripotency in these cells, umbilical cord or bone marrow stem cell, if they have to have the maintenance of the pluripotency, they also require the adequate and constant expression of lot of Wnt ligands as mentioned here, Wnt2, Wnt5a, Wnt4, and some of them, even if they are negative regulators, they make sure that they are produced in a constant rate and a constant balanced rate, not that the balance is skewed towards either activation side or an inhibition side. So this is also important to note. So coming back to this terminology, regenerative medicine, many of you would have heard about the terminology regenerative medicine, and it is coined in around 1992, and since then this field has gained a lot of popularity and it is increased because of lot of the potential the regenerative medicine has created lot of excitement and interest in the field of regenerative biology, and regenerative medicine is one of the major cause of driving the regeneration biology field. One of the most important reasons is designing of novel biological substitutes with the ability of the restoration and maintenance of normal function, which is the main scope of tissue engineering as the major component of regeneration medicine. If I tell you a person who have got insulin deficiency, type 1 diabetes, who don't have enough insulin producing cells in their body, which is supposed to be produced by the beta cells of the pancreas. Now they have maybe some autoimmune condition in which the beta cells are destroyed. So think about a situation where you can take out the cells from the same patient and you culture them in a petri dish, and you force them to become beta cells and you supply them back into the pancreas. Then that person will have normal insulin production. A simple solution to control his type 1 diabetes. So these are all the potential the regenerative medicine is offering. Tissue engineers combine cell transplantation, biomaterial science, life science principles, and mechanical engineering principles to construct new tissues for implantation. So, tissues are necessary for implantation. Like people have used, like sometimes the pig urinary bladder is being used to put into a human urinary bladder troubles. But the ideal scenario would be to make more of a humanized or human derived urinary bladder than using a donor animal's urinary bladder. Urinary bladder is not a complex tissue as such, but it's an important organ. If there is a hole or a damage is there, then they have to roam around with a urine bag. They cannot hold their urine. So one of the simple structure, people have done lots of transplantation of the urinary bladder in various developed countries. As it is inferred from its name, a cellular matrices consists of matrix with virus vectors or plasmid to secrete a specific growth factors or hormones in the extracellular matrix proteins inducing collagen, hyaluronic acid, and fibronectin to make sure that biocompatibility and without any cells that act binding to the host and the matrix cell interaction. One of the other approach what has been done is you take some people use some cotton candy, you may have seen it, that kind of matrix, or otherwise you take a donor organ, say heart or a kidney or something, and get rid of all the cells, decellularize it, and you got rid of all the cells. All you have now is a skeleton of extracellular matrix, ECM. Now you colonize the stem cells from the host or the patient derived stem cell, you colonize it into that matrix, so that now eventually the matrix will be dissolved by these host cells, and the new matrix will be secreted by these colonized cells, and so that this organ will become completely humanized, or a host derived organ you made outside the cell. So this is what you should keep in mind, the potential of the regenerative medicine. How Wind beta-catenin pathways comes into stem cell control. That is something which we should look into. Embryonic stem cell, we all know that respond to the activation of the Wind and beta-catenin signaling pathway. It is necessary, and if you put more of Wind3A protein, then it will stimulate, it will proliferate more and more. And it is important for the maintenance of pluripotency. Wind signaling is a must. Mouse embryonic stem cells are maintained by LIF. LIF stands for leukemia inhibitory factor, STAT3 signaling. Leukemia inhibitory factor and STAT, because normally these stem cells are vulnerable for transformation. It can become cancerous. You don't want that, so LIF comes into picture. Normally you have a feeder layer which secretes the LIF, or sometimes you have a feeder layer and you supplement the LIF from outside. Either way, leukemia inhibitory factor is necessary to maintain the stem cells. And LIF STAT3 signaling, STAT3 we have already discussed in the previous class, signaling through the transcriptional activation of MYC gene. MYC is a proto-oncogene, and MYC is a common transcriptional target of LIF and STAT3, and canonical Wind signaling cascades turn on this MYC genes. And MYC is also a very important gene for regeneration biology. Although MYC is normally connected to be associated with metastasis, cancer, etc., it is one of the pluripotency inducing factor as well. Because of MYC protein is further stabilized by canonical Wind signaling cascade, this pathway is necessary for the maintenance of undifferentiated mouse embryonic stem cells. Without this pathway, it will not hold back. Stem cells and Wind signaling in regenerative medicine, how is it contributing? Stem cells, including embryonic stem cells, bone marrow mesenchymal stem cell, it is also known as BMMSC, and umbilical cord derived mesenchymal cells, UCMSC, induced pluripotent cell, like where you can take the fibroblast and convert them into stem cell, that is iPSC, and other cells derived from fetal tissue or adult sources of bone marrow, fat, skin, etc. can act as the sources, have the future potential to be used therapeutically, because these cells can be maneuvered on a petri dish and you can make them suitable for transplantation, at least as a cell type. Mesenchymal stem cells such as BMMSC, that is derived from the bone marrow, and umbilical cord, they are somatic stem cells, are other most practical cell sources that can be used for regenerative medicine. These cells are found numerously present in a organism, and including the marrow, umbilical cord, blood, and adipose tissues. It is available, so there is no short supply of these cells, so you can access them easily and culture them. The MSCs, that is mesenchymal stem cells, differentiate into a wide range of cells such as osteocytes, fibroblasts, mesoderm-derived chondrocytes, myocytes, adipocytes, non-mesodermal derived hepatocytes, and even neurons. So maneuvering these cells have a huge potential when it comes to regeneration medicine. These cells can be isolated as a fraction of adherent bone marrow, so colony forming units, fibroblasts, also known as CFUF. And this umbilical cord mesenchymal cells, also similar to the bone marrow mesenchymal cells in case of gene expression profile. Although they are from two different sources, they have a lot in common when it comes to gene expression profile, which are they will be able to differentiate into adipocytes, osteoblasts, hepatocytes. Of course, you have to provide the ample environment for or ample stimulation to become, not that you take a umbilical cord cell and it will give only adipocytes, it will give only neurons, not like that. You have to give it the proper stimulation. So similar to embryonic stem cell, the regulation of proliferation and differentiation of these mesenchymal stem cells is also affected by Wnt beta-catenin signaling. No matter what is the source of these cells, they depend a lot on this Wnt beta-catenin signaling, and they are important in controlling the stem cell proliferation. Various studies have reported the involvement of Wnt beta-catenin signaling pathways. They are important in the murine and human embryonic stem cells maintenance. One of the most interesting factors of about the impact of the Wnt beta-catenin signaling on the embryonic stem cells have been achieved from the observation that the Wnt proteins are contributed in the maintenance of the pluripotency of, they are required for maintaining the pluripotency of the ESL, human and murine embryonic stem cell. The treatment period, what you fix for a given experimental regime, is an important factor that in the effect, what is the outcome you get it because of the Wnt proteins acting on the cell. Proteins in the ESC, embryonic stem cell pluripotency, such as some studies have shown that long-term treatment of embryonic stem cells with Wnt3a, that is the ligand of the Wnt signaling, caused differentiation towards a mesodermal lineage. Mesoderm means muscle, etc. So they are mesoderm derived embryonic origin. We will not go into the details of those tissues right now. In addition, there is also other studies. The previous research documents, they have indicated that the key components of the Wnt beta-catenin signaling, including Wnt3, 5, 5b, 7a, 7b, that list continues, and Frizzled receptors, ROR2, and they are expressed in the embryonic stem cell. That means they are waiting for a chance to interact with the respective ligand when available, either externally supplied or the cell itself that is secreting it. Embryonic stem cells respond to the activation of the Wnt beta-catenin signaling pathway in a very positive manner, especially maintaining the pluripotency, that is the vulnerability or the ability to differentiate into a given cell type. That is what we are referring to by pluripotency. Activation of the canonical Wnt signaling, Wnt beta-catenin signaling cascade is necessary for the maintenance of undifferentiated mouse such as bone marrow derived mesenchymal stem cells and umbilical cord derived mesenchymal stem cells. Are somatic cells are the most practical cell sources of regenerative medicine, because since their abundance is very high, picking them will not create any too much trouble to the host or the donor organism. The mesenchymal stem cells can differentiate into a wide range of cells such as osteocytes, fibroblasts, mesoderm-derived cells, myocytes, adipocytes, hepatocytes, and even neurons. Similar to embryonic stem cells, the regulation and proliferation of and the differentiation of these mesenchymal stem cells is also affected by Wnt beta-catenin signaling. So what we should understand is a huge number of studies have shown that the Wnt ligands are the pivotal roles that trigger, they play pivotal roles that trigger the Wnt signaling pathways such as Wnt2, Wnt4, Wnt5, etc., etc., and also its receptors known as frizzled 2, 3, 4, 5, and 6, etc. And also there are other related LRP co-receptors also comes into picture, and needless to say, the involvement of Wnt negative regulators also comes into picture such as WIF, DKK, and SFRP, etc. Furthermore, the canonical Wnt beta-catenin signaling pathways, they have a substantial role that played in the maintenance and self-renewing of these cells throughout until they are waiting for a signal for differentiation. Until then, the Wnt signaling has to be turned on and active in a prudent state. Due to increase in self-renewal potential and decrease in the apoptosis, cell culture treatment with Wnt3A. Normally, all these studies people use Wnt3A because it is more active on the cells. So it doesn't mean that Wnt4 is not good or Wnt2 is not good. People usually use Wnt3A, enhance the multipotential population of MSCs as well as human adipose derived stem cell. Additionally, the overexpression of LRP5, which is the co-receptor of result, LRP5 has been shown to increase the proliferation rate of mesenchymal stem cell. In contrast to Wnt3A, Wnt5A, if provided, it inhibited the proliferation of mesenchymal stem cells by activating a non-canonical pathway. It still binds to the receptor, but compared to Wnt3A, Wnt5A can trigger a non-canonical pathway. The dual effect, what you see in Wnt beta-catenin signaling, in which Wnt are increased in the mesenchymal stem cell proliferation rate at low dose. At very low dose, it is increasing the proliferation rate while suppressing the proliferation rate at a high dose, reveal that the intensity of Wnt signals can result in various or even opposite biological function. So you need food for you to live properly, but if you are overdosed with the food, you will end up in getting hospital bed despite having an idea that food is a necessary molecule. So this logic you should have in your mind. With signaling in the cardiac regeneration, let us see how cardiac regeneration is influenced by Wnt signaling. Reactivation of Wnt beta-catenin signaling following cardiac injury is shown to have pivotal impacts in cardiac progenitors in several ways. Many research authors have reported that beta-catenin depletion induced recovery in a resident cardiac progenitor cell population exhibiting alpha MSC promoter activity and expression of cardiac transcription factors such as GATA4, which is a cardiac specific transcription factor, and TBX5. After infarction, they are turned on. This effects was mediated by enhancing cardiomyocyte differentiation of alpha MHC, major histocompatibility complex, plus SCA1 plus cardiac progenitors. So there is a congregation of these three together. And also the stabilization of beta-catenin by pharmacological methods using the same promoter decreased the cardiomyocyte differentiation of this cardiac progenitors. So we can understand how the Wnt signaling is influencing the cardiac repair or cardiac regeneration. Knockdown of another protein called KLF15, which is a Krepel-like family protein transcription factor, which is a negative regulator of beta-catenin signaling, TCF transcriptional activity, resulted in cardiac beta-catenin transcriptional activation along with functional cardiac deterioration in normal homeostasis. No need of an injury. Even in normal situation, if you knock down the KLF15 and it can often lead to hypertrophy. So homeostasis condition also, by tweaking the Wnt signaling, you will end up getting often some very dramatic effects. KLF15 deletion induced endothelial lineage differentiation of cardiac progenitor cells. They concluded, the researchers who discovered it, they concluded that KLF15 controls the cardiac progenitor cell homeostasis in the adult heart, similar to embryonic cardiogenesis when the heart is being formed via the inhibition of beta-catenin transcription. So KLF15 have a significant role in controlling or maintaining the level of the beta-catenin in a threshold level. The treatment of SCA1 and also the promoter of Wnt inhibitor together, and also the secreted frizzled related protein, that's SFRP, which is a negative regulator of Wnt signaling. It is secreted from the cells. It will go and sequester the Wnt ligands. So before the Wnt finds the frizzled receptor, actual frizzled receptor, this SFRP will fool the Wnt and act like a sponge. It will get rid of how sponge is removing water. The Wnt will be removed by SFRP. They promote differentiation of the cells after ischemia perfusion injury. In other words, what we understand is the treatment of the progenitor cells with SFRP, which is a negative regulator of Wnt signaling, suppressed their proliferation and primed them for cardiac differentiation. That means you will end up getting more of a cardiac type of cells if you tweak the SFRP, if you deliver more of the SFRP, which is a negative regulator of Wnt signaling. Beta-catenin overexpression and consequently the activation of Wnt beta-catenin pathway decreased myocardial infarct size through the differentiation of fibroblast into myofibroblast and resulted in slight improvement of cardiac function and decrease the infarct. Infarct, what basically mean is that fibroid tissue that is formed after myocardial infarction. So Wnt signaling leads to differentiation of epicardial cells into fibroblast via the process of epithelial to mesenchymal transition. So what is its role, Wnt signaling, on lung regeneration? Increased Wnt beta-catenin signaling may be contributed to epithelial cell injury and hyperplasia usually, as well as impaired epithelial to mesenchymal crosstalk in idiopathic pulmonary fibrosis, which is a condition which normally get fibroid lungs, which can reduce the functioning of the lungs. Canonical beta-catenin, Wnt beta-catenin signaling is activated during lung epithelial regeneration and regulates bronchovascular system, that BASC cells proliferation after lung injury. So the regeneration of the injured lung seems to be insufficient to repulse the fibrotic disease. So Wnt signaling can play very interesting roles in kidney regeneration. So this is slightly different and opposite to what you have seen so far. When macrophages are depleted from the injured kidney, the canonical Wnt pathway responses in the kidney epithelial cells are decreased, and the administration of the Wnt pathway negative regulator DKK2, it increased the repair process. That means kidney has got a more of a Wnt signaling by default, so you want to decrease it. Activation of beta-catenin by Wnt1 or stabilization of beta-catenin protected tubular epithelial cells from apoptosis. Hence, the endogenous beta-catenin is substantial for renal tubular protection via acute kidney injury by provoking the cell survival through multiple mechanisms. So now if you look into the bone regeneration, the gain of function of Wnt beta-catenin signaling pathway resulted in high bone mass in mouse models, and therefore, not surprisingly, in mice models with an activated form of beta-catenin in osteoblast, that is the bone cells which gives rise to the bone, and the knockout of axin-2 have considerable increase in bone healing. So the administration of recombinant Wnt3a and also lithium chloride can increase the healing potential following the start of treatment after the bone fracture. This has been demonstrated in the mice. Wnt responding cells which are undergoing a transient step cell differentiation induced by local Wnt stimuli can increase or restore the regenerative capacity of the bone. So we have seen how important the Wnt signaling contribute to different tissue regeneration and repair process. We'll study more about the other signaling events in the next class. Thank you.